One or two to let you know.
Okay, good morning. Good morning from Montefiore at the Bronx, all of you on Zoom. We have really a great treat today by having Dr. John Greally here, who is here at Montefiore, Professor of Genetics and Pediatrics at the Albert Einstein College of Medicine, and also has an affiliation with the New York Genome Center. Dr. Greally is going to speak to us about cardiac genetics in the Bronx, and I do like the title, How What's Rare Globally Can Be Common Locally, which is a really nice introduction to our highly diverse borough here in New York City. But Dr. Greally was not born here. He himself is from Galway, Ireland, where he also completed his training in medicine, then moved to the United States and trained at the University Hospital of Pittsburgh and at Yale University in clinical genetics, which has been his focus ever since. He's highly published, over 210 peer-reviewed publications, and has multiple grants from the National Institutes of Health, which hopefully are holding up during this difficult time. He also serves clinically as a professor on the genetics service at Montefiore Medical Center. And today will introduce us to, I think, only recently funded New York Center for Rare Diseases, of which he is the executive director. He's also the founding director of the Center for Epigenomics at Einstein, and his book, I'm going to make a pitch for that, his book on epigenetics, History, Molecules, and Diseases, will be published this year. So we're really excited to have you here. Also somebody that's just across the street from us in the other building, we always reach out to for clinical problems. So I'm very happy to welcome you today and look forward to your lecture. Thank you, John.
Thank you very much. So it's April 1st and you can't imagine the temptation, but I am not going, it's too early in the day. So everything in this is going to be factually accurate. So good morning, everybody. Thank you for the kind introduction. And I'm going to, this part about what's rare globally can be common locally. I'm going to make sure you stay attentive because that comes at the very end, and it's good. So, but I do want to talk about what we're doing here in the Bronx, in particular at this New York Center for Rare Diseases. Quick conflict disclosure, nothing to disclose. And I do want to acknowledge, sometimes get slides from colleagues, and I can't not acknowledge Diane Milowitz, who's world expert on thoracic aortic disease. So I'll be showing something first. So this, I want you to see the kind of pitch that I make outside the Bronx about medical genomics in the Bronx. My point about the Bronx is that it's difficult to do medical. If we build systems that work here, they're going to be resilient and universal. So why is that? Well, we know these things, but I'm showing you what I show to other people. If you look at the demographic categories that people tick off in the US Census, 91% of the Bronx claims an ancestry that is other than European. So that makes us distinct. And this is not going to get any less over time because there's a disproportionate number of individuals who are European white in the older age. So we're going to become more, not less diverse. I know we're not allowed to use the word diverse, but just between you. When I tell people that the Congressional District 15 in the South Bronx is the poorest congressional district in the United States, their jaws drop. They don't understand that 36% of people could be living in poverty there, and 26% are so, like myself, but different immigration circumstances, obviously 34% of the Bronx residents have immigrated. These are astounding statistics for people who come from relatively more affluent populations. And I love this map here. It shows the darker blue is the Spanish-speaking southern part of the Bronx. The lighter blue is the predominantly English-speaking northern part of the Bronx. But if you take away those two languages, yellow as the third most commonly spoken language are West African. So this reflects the enormous diversity of immigrant communities. And what I point out to people is that we have the resources of top-class health system and a research-intensive medical school embedded in this community. And that's unusual if you think about it in terms of our peers around. One of the things that's critical, not just for us delivering care in the health system, but also for us doing research in medical school, is that we maintain those community relationships, because if we screw up with those, we're screwed. So we have to be, this has led to the realization that we have to be very careful, especially with things like genetic information. Because if you think of how genetic information is received casually by people that we serve, a lot of it has to do with paternity testing and criminology, as opposed to the potential for doing good. That's their health. So what I like is that I'm held to a high standard by my patients. They are appropriately suspicious of the harm that could be done with genetic information. And I think that their level of concern is much more appropriate than some of the more demographically white and perhaps socioeconomically more affluent communities around the country who are probably too trusting of their information. So I think we have it right. We have started to develop principles for how we should be having oversight of our genomics research. I'm not going to go through these in detail, but this one at the top is very important. And we saw a lot of it during COVID, where people were dying disproportionately in Black and brown neighborhoods. And a lot of well-meaning colleagues around the country approached me and said, well, we'll sequence your patients in Bronx for you. And they had made the implicit and racist assumption that it was because the DNA was Black or brown, and that was fundamentally what was going on, as opposed to being afraid of ICE agents outside mine. The other thing that I will highlight here is principle number 4, which is that at the end of the day, what we do in terms of our research should be coming back in terms of care. If it's not embedded in EPIC at the end of the day, so we can use it as a guide to our patients, we're probably not delivering the value that our communities. Now, these are principles that have been developed by basically white men working in Montefiore and Einstein, and these are therefore draft. We want them to be reflected back by the communities that we serve, so they become a mature document that has buy-in from. Because of the fact that I've been influenced by these conversations, I was invited to participate in March of last year in a World Health Organization meeting in Geneva, where we talked about principles for genomic data, privacy, and sharing, which are two sides of the same coin. And because of the fact that I was kind of out there, in my views, I was one of 2 people from America who were invited to speak. Everyone else around the planet talks about how there has to be trust between the care provider or the researcher and the person who's participating in the study. And I said, that doesn't work in the Bronx. You have to give the participant or patient control of their data. And the fact that in this highly wordsmithed document, the word control ended up being in the final version was a victory. One of the things that we're doing at the moment in terms of research is, with really generous help from the Department of Pathology, we're getting 3,000 de-identified blood samples that represent the 25 zip codes. And what we're going to do is we're going to do sequencing on, which will help us to understand 2 things. Where on planet Earth did people in the Bronx originally come from? And how have their genomes become admixed, or basically blended, as people have intermarried across ethnic and geographic groups? And this will help us not just to understand things from a superficial point of view, but we will then know, and this will become relevant at the end of the talk, where within the Bronx we might be seeing certain types of diseases overrepresented, which we can then push out to the primary care providers and say, if you see this type of condition in somebody who may have this ancestry, think of this rare disease. We'll get to that at the end. One of the things that I'm doing is trying, as a geneticist, is trying to solve rare diseases. The dirty secret about rare disease genomics is that even when we sequence every nucleotide in the genome with current technologies, our diagnostic rates are low. We'll get more into that time. But in this particular study that we did, our 388 patients from Montefiore and Einstein that we sequenced, only 24%-ish were positive. That is a very low diagnostic rate. And when I talk to peers around the country, they tend to talk about higher diagnostic rates that they get for equivalent disease presentations. So I was wondering, why would that be the case? This paper came out in the New England Journal of Medicine. I think it has a lot of lessons for us here in the Bronx. This is a study of the United Kingdom and Ireland, and I'll remind people that Ireland is a separate and independent jurisdiction. We got our independence. And what they did was they looked at 13,500 individuals with rare diseases. And when they were having difficulty making diagnoses, they tracked back to what those families. And on the right are the big things that they were pointing to. An inability to test a trio, in other words, both biological parents and the affected individual. If you think about the Bronx, that unstable families characterize many people as their prematurity, which is high frequency in Bronx. Exposure to anticonvulsants is, I think, probably similar in all patient populations. But maternal diabetes, and they pointed out Black or African ancestry. Think of all these things. These are all factors that are going to be prevalent in the Bronx. It's one reason why we have greater challenge to make diagnoses here. The other thing that I sometimes wonder about is, in terms of making a difference in a patient's life, if we're doing a $1,000 test to try and diagnose a rare disease, would we be actually better off just getting $1,000? And this is where it impacts the work of Kevin Fiore in pediatrics, who's been working a lot to create what we all see in EPIC when we look at the bottom left corner, social determinants of health. So as we're offering these highly sophisticated tests for rare diseases, we have to bear in mind that people's lives are complicated, and we have a lot of social determinants of health that are impacting patients. And as people are trying to receive this complex information, how are we intersecting other pressures in their life? So this led me to pitch the idea in the early post-pandemic period when I was frustrated about what we were not doing well with genomics, medical genomics delivery in our health system, to set up the idea of calling ourselves the New York Center for Rare Diseases. I'm not sure that my colleagues downtown in Manhattan are happy that we have taken this in the Bronx as our name, but I think we actually do rare diseases better than elsewhere, and I'll tell you why. It's because we're doing this at the health system scale. Everywhere else where you see rare disease services being provided tends to be kind of mom and pop shop operations. Somebody's really good at Fanconi anemia, so they have a world-beating clinic there, but it's one person, one service, one lab. What we're trying to do is to make sure that this can happen at the level of the entire health system. And there are three components to it: the excellence in care, innovative therapeutics, and advanced diagnostics. Where we start with is care. There are about 40 rare disease clinics at Montefiore right now. That's extraordinary. And the ones that I've highlighted in boxes here are the cardiac clinics. You can see cardiac amyloidosis, sarcoidosis. You can see the Center for Cardiogenetics. I run the Paul Coors Aortic Clinic, where I see patients referred for cardiothoracic surgery. And we also have a pediatric program that Nicole's up. So this is excellent. But the question was, how can we do better with diagnostics, which is what I really do on the research side. So I went out and I pitched partners. And I said, we have no money here in the Bronx, so you're going to have to pay for all of this, but this is what you're going to get in return. You're going to get access to patient genomes, which are incredibly challenging. The Bronx, I said, was the crucible for medical genomics. So anything that we forge here is going to be universally resilient. I wouldn't be telling you this story unless I was successful, because GeneDx, the diagnostic, PacBio, and Google Health all pitched in to the tune of hundreds of thousands of dollars for a pilot project, which is now expanding to a broader project over the next 3 years. We'll be looking at 1,200 families. We were also awarded Center of Excellence status by the National Organization for Rare Diseases. And as I said, we did a kind of a punk rock, called ourselves the New York Center for Rare Diseases. So we are also trying to develop a track record in therapeutics. This is where we have the most ground to make up right now. I have a patient who has a neurodegenerative condition, a little 4-year-old Venezuelan kid, and the condition is called Pelizaeus-Merzbacher syndrome. And it looks as if this should be something where fix his splicing defect that is causing his problem. If we can use these so-called allele-specific oligonucleotides, infuse them intrathecally and periodically, and slow down the rate of progression of this condition in its brain. We're also developing, and this is being funded by the Chan Zuckerberg Initiative, yes, that's Zuckerberg, and the N. Lorem Foundation, who create these ASOs for therapy. We're also developing a relationship with the New York Stem Cell Foundation. We're extraordinarily good with stem cell modeling for diseases. Then the question is, I haven't mentioned the medical school up to now. Medical school is the great untapped resource. Very, very good researchers over there. very little track record of rare disease research, nothing really in the therapeutics area. And you can see one cardiac program here, Bernice Morrow's study of DeGeorge syndrome. But this is a pretty sparse representation of research in a medical school focused on rare disease. We can and should be doing much better. But ultimately, it should be an integrated ecosystem where what we're doing on the clinical side feeds over to the basic science side of medical school. Okay, now I am envious of all of you in adult cardiology because you have high diagnostic rates. You have higher diagnostic rates than I get when I see kids and biologists. I'm showing here a very, very quick literature review. This is 100%, and as you can see, the proportion of individuals, all comers who walk in, cardiomyopathies, arrhythmias, or thoracic aortopathies, have relatively high diagnostic rates. If you get a 20 to 30% diagnostic rate, it's really worth sequencing those patients. And I'll go into more detail with the aortopathy issue because while this at the bottom looks very impressive, it's a real outlier. So why do we do genetic testing? Like, what's the point? Part of it is if a family wants to understand, is they've just had a child to understand the risk of future pregnancy, because that's information. Just understanding why did it happen to them? And often, as you'll see with most parents, they will blame themselves for a child's problem. This is very much beyond. If we can make a diagnosis of rare disease, all of a sudden they can talk to other families around the United States, around the world, who have a child with the same condition. That's enormously supportive. But cascade testing is something which can apply not just in the pediatrics realm, but in the adult realm. And this is an example here of somebody who presented in a 4-generation family who had thoracic aortic aneurysm. There's one other person known to have it in an uncle. And everyone, where you can see that little red star, which is just about visible on the screen, when they found the variant, the mutation that was causing the problem in the proband over here, they saw that a whole bunch of other people were carrying it. So what's the next thing you do? You image the thoracic aorta, and all of a sudden you went from 2 people recognized to have aortic aneurysms to 14. That's hugely important because there's significant risk of sudden death in these individuals. And some of these people here, the generations further down the chart, are too young to develop thoracic aortic disease at this point. But if you screen them, some of them are going to probably turn positive. This is another example of the value of cascade screening. This is the ACTA2 gene, which can cause thoracic aortic aneurysms. And as you can see, when you look at this 4-generation family, while quite a few, shown in orange, have the variant, only about half of them have thoracic aortic aneurysms. But if you then ask, the 2 variants are also associated with coronary artery disease. If you add in the people with coronary artery disease, all of a sudden, everybody carrying the variant has something. The aortic issue or coronary artery disease. So this is the principal values of being able to do cascade testing. You know who else in the family is at risk. You should screen, because you have the chance for an earlier intervention. I feel uncomfortable giving this slide because I'm not going to start talking to cardiology colleagues about the types of drugs that are used in cardiac conditions. But these are the ones which will jump out when you start asking the question about what are the therapies that you can provide based on certain genetic variants that have been identified. And this is another value of what we can do with genetic testing, because if you can put people into these groups, then you can have the appropriate therapy for them. And I'm particularly interested in the cardiac myosin inhibitors at the end of the talk, because that's what's really relevant to this population of Bronx that I want to highlight. Focusing, as I tend to, on thoracic aortic disease, the gene that's involved can be helpful in determining how you should manage the patient. So there are some genes where you know that the rate of dilation of the aorta is going to accelerate. So you get in there earlier and you replace the affected part of the aorta. And the TGF-beta receptors 1 and 2 and SMAD3, which are associated with Loeys-Dietz syndrome, are much, they're concerned. If if you see that in a patient, get in there quickly, even if their aorta is only 4.1 centimeters. There, you know, if you start looking at the probability of an aortic event, which in this case is surgery or a type A or type B dissection, some genes are much more likely to cause problems than others. One in red here is MYLK. Again, you see the gene and you're able to say more concerned, less concerned. It helps with the management process. And even the variant within the gene. So this is ACTA2, which I was talking about before. And if you have a variant at the arginine 179 position, sorry to get technical, but if you find that the problem is at that position, get in there early, because these people, you can see at the age of 10, they are getting events. And they can end up requiring replacement of their entire aorta because of this fragility. Missense variants, where you replace one amino acid for another, as opposed to killing the gene itself. Strangely enough, a missense variant in MYLK is much more damaging than when you lose the function of the gene altogether, and so on. So that sounds great, but the problem is that the rate of diagnosis in aortic disease is awful. And I'll zoom in and show you what's going on here. Well, you know, went through medical school. You saw all this stuff about Marfan syndrome, how we need to be able to pick that up. And yes, that is definitely worthwhile. But only 5% of people with thoracic aortic aneurysms or dissections have a syndromic presentation, 95%. If you look at the syndromic patients, I love it when I can diagnose a syndromic case because my diagnostic rate is about 50 to 60%. But it's only 5% of individuals. And the ones that we're really worried about, ones that look like people in this room, people who have no obvious syndromic features, but could have a 4.8 centimeter aortic root. If it's a familial situation, where if there was an uncle, there was a parent, whatever it might be, who probably had the same condition, those are ones that give us a clue, at least from family history. So it allows us to get in there and do the testing a little bit quicker than we might have with other patients. But it's still a pretty low diagnostic rate. That's probably close to 10%. But the ones that are sporadic, there's no other family member, it's lower. So these are the ones we need to diagnose. And probably what the future is going to bring is where we are able to offer genetic testing pretty liberally to all sorts of people, not just to cardiology, but to any clinic, so that we can get to these conditions. I need to persuade you that I'm smart. So what I'm going to do for the next few minutes is I'm going to talk to you about new technologies that we're using at the Eurocenter for Rare Diseases that allow us to improve current genomic diagnostics. This is the simplest that I could make it. Currently, when we do even the most sophisticated sequencing, the length of DNA that we sequence at a time is 150 base pairs. That's what's clinically approved. But what's coming through on a research level is what's called long read sequencing. It's aptly named because it's 15,000 base pairs. That's 100 times longer. So why is that valuable? If I were to take each of your genomes and ask the question, how often do I have to walk along your genome before I see a variant that distinguishes you from the reference human genome or the two chromosomes that you have, which is another comparison? It's not every 800 to 1,000 base pairs. Okay, there's going to be some sort of variant. So if you have if you've got a 150 base pair read, it's probably not going to have a variant. Have it maybe one time in every 5 or so. But in a 15,000 base pair read, you're going to have 10, 15, 20 variants in that region, which then allows you to stick together the reads that are coming from one chromosome versus another, which is what we call phasing or resolution of haplotypes. The average length of a gene in the human genome is 27,000 base pairs. So it's pretty soon we're able to say, this is one copy of the gene, this is the other copy of the gene. And a problem that can occur with the recessive condition is you can say the patient has 2 areas of damage within the gene, but you don't know whether those 2 areas of damage are on the paternal chromosome and the mother's chromosome is absolutely fine, and therefore they're just a carrier and they should be okay, or whether there's damage on both chromosomes, in which case one on each chromosome, and that therefore they should be affected. So long read sequencing is helpful with that. Long read sequencing just gives you more of the genome. The GBA gene is the gene that causes Gaucher disease. It's a common enough problem. It's increased frequency in individuals with Ashkenazi and Jewish genetic ancestry. But short read sequencing, very little secret, it really doesn't cover the end of this gene. Whereas long read sequencing just walks all the way through this without a problem. This is an example of how you can find structural variants. In other words, where the chromosome breaks or rearranges in some way, in a way that you just can't do with these 150 base pair sequences. And this is a really weird example where we have little virus-like sequences in our genomes, and they can pop out and land in a different place. One of them is called an ALU element. And this is an example where an ALU is present on one chromosome here, but not present on the other. This is an example of something which we have chronically underestimated as a cause of disease in the genome, and it's a repeat expansion. Very difficult to test unless you do a very specific assay, you know where to look. But long-read sequencing allows us to read through these variants, which are, in this case here, it's a myotonic dystrophy locus, where there's CTG, which is repeated CTG, CTG, CTG. And all of us have a certain number of repeats in tandem at this particular locus. And this would be an example of the chromosome where it's present in a normal number. When you see these numbers here, 4,824 divided by 3 is about 1,600. There are 1,600 of these CTGs on this particular chromosome. But it's not sitting in a coding sequence. It's sitting at the end of this gene here and upstream from this gene here. So how does it cause problems? What you're seeing in blue here is unmethylated DNA. Red is methylated DNA. Unmethylated DNA is the loci in the genome that regulate how genes switch on and off. In this case here, it's right at the beginning of the SIX5 gene. And this is the normal promoter of this gene. But when you expand a repetitive sequence, you see how everything nearby has now turned red? That's because the DNA methylation has encroached on this promoter sequence. It's shut the gene down. And it's because of that that you get myotonic dystrophy, not because of mutation. This is an example of cancer sequencing, but there are obvious things that can be brought over to rare diseases. In cancer, prostate cancer in particular, you see fusion genes where you get the beginning of one gene, the end of another gene. And this can be an oncogenic driver event. This is a well-known gene in prostate cancer, SKIL and SLIC45A3. And what you're seeing is the blue gene is now fused to the orange gene. And so you can see this because of long read sequencing of the RNA, not just of the DNA. So we're going to be using this in New York Center for Rare Diseases, not really to look for fusion genes, because those are more oncogenic events, not associated with rare diseases, but instead to look to see if there are problems with splicing, which are often very difficult to predict with DNA sequence. We have imprinted loci in the genome, where it is unmethylated in one chromosome, methylated in the other. That's what's presumably happening in everybody here. But sometimes we see there's loss of imprinting, and this can be associated in this case here with transient neonatal diabetes mellitus of the PLAGL1 locus. So again, that's something else we can use in the stop. So here's the future of our testing. It's what I call knucklehead testing. Think about it. If I have to think through doing 5 separate tests to try to understand what's going on with my patient, but with long read sequencing, I get all of it in one test. It's just a matter of processing the data. Even if I don't have a suspicion for disorder, it's there anyway. We're going to be able to, I would order that test, assuming that it's the same cost. It'll just give me more information. So that's the future for genetic testing. When the long-read sequencing technologies become qualified, part of the reason why our partners want to work with us is if we can show that we're picking up diagnoses with long-read sequencing in our Bronx patients, it's going to help them make the case that this should be approved as a laboratory development. Again, to show you that I'm justifying my paycheck with the Einstein set. One thing that I'm interested in, this question of epigenetics, the introduction to about how I've written a book on. 98% of our DNA does not code genes. And the 98% is where all the regulatory loci, like that blue unmethylated promoter, those those loci are really important in terms of switching genes off and on. The question is, can they cause rare diseases? The answer is definitely they can. And there are some well-known examples where the gene itself, like PAX6, is located distant from a mutation. And this mutation is happening at an enhancer of expression of PAX6. And if you have a mutation in that enhancer, you can have aniridia. You can have PRO-bound sequence where the jaw is so small that during embryogenesis, the tongue pushes up in the mouth and prevents your palate from closing. So the combination of small jaw and cleft palate is the PRO-bound sequence. And then you can have skeletal abnormalities as well. So we know from rare disease studies, it's isolated instances that these non-coding 98% of the genome sequences are potentially causing diseases. But these are really dramatic examples. How can we pick up more subtleties? So I showed you this blue and red before. At a non-imprinted locus, you would expect everything on one chromosome to look like everything on the other chromosome. So if it's red, it should be shared. Blue, it should be shared. But something funny is happening here. As you can see, something different that's going on. And what is actually happening here is that there is a sequence variant that's present on this chromosome that is causing the region to be unmethylated blue. And the lack of that variant on the other chromosome is allowing it to become methylated. So what does this variant do? This variant introduces a C to the position, which restores a transcription factor binding site, something which binds to the DNA, helps it to open up, become active. And in fact, calling it the alternative or the less common allele is a bit of a misnomer, because that is the case. You can see in gold the C individuals who are in a minority in Africa and in South Asia, but in the rest of the world, C is actually a more prevalent allele. And if you look at our more recent cousins in the primate lineages, you can see that the C is the ancestral allele, this locus. So the question is, can we identify these loci at scale and look in patients with rare diseases to see whether they have abnormalities in these loci? And this is a very psychedelic looking image, I know. But what David Yang in my group is doing is he's partitioning the genome into these blues and reds, and red is the majority. He's looking for outliers. So he's looking for loci which are typically red, but occasionally blue, or typically blue and occasionally red. Which, if you find that and it's happening near a gene that's plausibly associated with the condition patient, that could be a candidate for causing. So this blue-red mix on the right here is just when the event is a bit more common and less likely to cause rare disease. And so this is an example of David is now stacking up hundreds of these alleles with this allele stacker approach. And we have a collaboration with Google DeepMind, who people got the Nobel Prize last year, because what they have been working on is proteins. But what Google DeepMind wants is to have the equivalent for looking at the 98% of the genome and seeing what is predictably causing problems. And so we're very excited by that. And the question, of course, will be, if we do start finding these things and they look like they're plausible, can we get them into clinical reports? That is a challenge because the field is very conservative. As you can see, I've been involved with some discussions talking about how we can move this forward. I hope that we can become persuasive. I'm going to mention advanced phenotyping. So phenotyping is, what does the patient actually look like? And that information is typically very poorly provided by us as clinicians to the diagnostic lab. They typically say, the kid has seizures in a small jaw. You have very little for the lab to work with. Can we do better? So there are two places where we can do better. One is looking at the cells from the patient. The other is describing the patient. So Dinah Milowitz, as acknowledged at the outset, has been taking this challenge of diagnostics, classic aortic disease, and really trying to figure out how we can do that. One of the things that she's been focusing on is how splice variants, in other words, a variant which is present close to the splice site of the gene, either the splice receptor, splice donor, the exon. The variant there is very, very difficult to interpret, but her evidence is showing that it's probably the case that a lot of the diagnostic yield that we're missing is mediated by variants of those loci. So question is, how do you test that? You know, prediction is all very well, but you're not going to base care on something which is so research based. So we have to basically look at cells from the patient and see, is the splicing actually effective? And this is a picture from the GTEx resource. I'm looking at FBN1, which is the gene for Marfan syndrome. And the problem is FBN1 is just not expressed in whole blood. So we can't just take the RNA from the patient's whole blood and test it. We have to do something else. As it turns out, fibroblasts, which are over, I can't remember there. Anyway, fibroblasts have a very high level of expression of FBN1. So skin biopsies may be the way to go with these patients instead. The other thing is, if we need to look at the cells that are mediating the disease, and not just looking at peripheral blood, the great thing about aortic disease, and to some extent with other cardiac conditions, is that we can get the cell types that are mediating the disease because there's a procedure, there's a biopsy, there's a surgical resection of the aorta. And this is where we're starting to work on developing a program that will allow us to study things like splicing, DNA methylation, and so on. These tissues here, it's an enormous opportunity for us. For some reason, people aren't looking at it. What I'm showing here is the beginning of the FBN1 gene, which starts on your right and proceeds to the left. And everywhere where there's a sort of a larger vertical tick is one of the protein coding exons of the gene. But what I'm showing below are the regulatory loci, the loci that help the gene to switch on and off. So my big question is, what happens, especially when you're close to the promoter, if you have a DNA sequence variant which is present on these loci? And because we can take the aorta, we can do this long read sequencing, we can look at the DNA methylation, we can look and see, like in the example of that locus that I showed you with the PU1 binding site, we can see whether there's a difference in DNA methylation between the two alleles, because these things are typical, rare variants and heterozygotes. So this is a project that has funding from the Paul Coors MD Foundation, working with Joe DeRose from cardiac surgery, and Corey Hirschenhaus, extraordinarily good resident working with the DeRose team. He's going to be spending a year working in the lab, getting these samples, preparing the tissues, allowing us to set up to do the sequencing that we need to do. This is a model I'd like to see extended further, cardiology and other areas as well. So the second type of phenotype, that's cellular, is just describing the patient better. And we have actually a very good medical informatics team here at Montefiore, the Center for Health Data Innovations. What we're able to do is an artificial intelligence approach. Now, you can't have a talk in 2025 without saying the words artificial intelligence. So just being pretty generic there. But there's an approach called natural language processing. What you can do is you can look into the notes that we have from our patients, and we can pull out these words, and we can ask the question, do these correspond to human phenotype ontology terms? And human phenotype ontology means that if one person says the patient is obese, another person says overweight, another person says like fat, you can condense it down to one single term that is machine readable. In other words, the computer knows that if it has HP, blah, 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 it is something that is associated with a certain gene disease. So this is turning our notes into something that a computer can understand. Why do we want to do that? As I say, we typically provide very little information about patients. But the more information we provide, the higher the diagnostic rate. So we're going to start, our eventual goal, once we know that we've got the HPO natural language processing working, is to mine every note in Monte. So we'll have an HPO list for every patient. And then if we need to do genetic testing, it's right there. So we can send it to the lab. This is where we are going to address another problem that we have, which is that physicians get a report back. The report is typically unreadable, and we don't have any input into the diagnostic process. So that's why we developed genome diaries. So this this is a way of entering information about a patient. In this case, I'm asking it to find a single transverse common crease, so we can add it to the information that's present in the patient. When I look down, there's no genetic disease that's lighting up, but I realized that from a CTA or something like that, there's arterial tortuosity. I add that in, and you can see that all of a sudden Lowenstein syndrome type 2 comes. So I really like the dynamic nature to this, because what it allows us to do is say, I'm going to put this in, oh, that had a big effect, but I'm not really that confident in the terms. You pull it back out again and other things. And it really is what it's doing is it's looking at not just the information you're putting in, but the patient's genome. And it allows you to tell whether something looks like it's really close. Okay, you've been very patient. Now we're going to talk about rare diseases in communities. And this is the punchline for today. So Shri Raj, my colleague in genetics, is very unusual in that she is combining two areas of interest. One is population genetics, the other is rare diseases. Most people who study population genetics, where they look at the structure of populations, the ancestry, the relatedness, because they're trying to draw inferences about conditions, they look at common diseases, look at coronary artery disease, or asthma. What Shri and her extraordinarily talented postdoc, Mariko Shiki, have done is they've tried to understand rare diseases. So this is the results of their first study, looking at 25,000 people in New York City. And what we're completing at the moment is a second paper where they look at 245,000 people in the United States. So this is a preview, but it's also local, parochial New York City. So what they did was, I want you to imagine. So I'm going to take you two and myself. Turns out yourself and myself share a block of DNA on chromosome 6. Then when I look at my DNA and I look at chromosome 14, I share a block of DNA on chromosome 14 with you. But when we look at your chromosome 12, you have a block of DNA that shares back with you. So we're not first cousins or second cousins, but what we've realized is that we are from the same genetic community. There's an interrelatedness that we have because our people have basically intermarried for a while. So all of these circles here represent these identity by descent, or IBD group. And if it has a dark circle around it, it means that that group is so interrelated that they qualify as a founder population. In other words, a relatively small number of people gave rise to today's modern. And of course, the first one that we would be familiar with as geneticists, because we offer genetic testing specifically for Ashkenazi Jewish individuals, it is Ashkenazi Jewish. And when we looked, Shri and Mariko, I should say, looked in this group, they looked to see what rare variants are overrepresented in Ashkenazi Jews. And we saw the things we'd expect. There's Tay-Sachs disease right there. Familial dysautonomia is one that we know about. There's and if it's got a number here, that's a PubMed ID. It's been published. It's known. So that was a good test for us to say that we were getting the results we would expect. We then focused on Puerto Ricans. If I see a kid in my clinic who's got albinism and he's Puerto Rican, I don't think of the typical tyrosinase pathway issues. I think of Hermansky blood bugs because I work in the Bronx and I understand that that's what we've been treating here in the Bronx for generations. And the top of the list here is Hermansky blood bugs that are type 1. And this 0.097, which is about 0.01, that's 1% of all of the chromosomes of the Puerto Rican population studied have this variant. So since we have 2 chromosomes each, that means 2% of Puerto Ricans are walking around with this a recessive condition. And while many of these conditions have previously been described, we also, through Shri's approach, managed to find new rare diseases that have not been previously identified and in Puerto Ricans. And obviously, David Lowe would love to know about bone marrow failure syndrome 2 mediated by this gene variant. So this is an example of how we can push this out. I need to tell you about the Garifuna. Hands up who knows about the Garifuna. Okay, this is typical. There are 200,000 Garifuna in the Bronx. We see them probably every week, and nobody knows their story. So the story of the Garifunas from the 1500s to the 1700s, they represented a group of escaped formerly enslaved Black African ancestry individuals who typically escape from somewhere like Barbados. The prevailing winds come from the Atlantic and they would blow. If you could get onto something that floated, you'd have a chance of ending up at St. Vincent. So the escaped African individuals started to accumulate on St. Vincent, where they met and intermarried with the indigenous Arawak and Carib communities. And they stayed as a community for a couple of hundred years until the British, also my own colonial oppressor, came along, decided they wanted to rid the island of these inconvenient people. And they basically had battles against them, killed a lot of these Black Arabs, as they call them, and took the rest of them, evacuated them by ship to Central America, where most of them ended up in Honduras and to some extent Belize. Because they'd been living independently for centuries, they were able to reestablish communities quickly in Central America, and they expanded as a population again. And now they have had migration to the United States, as many people do from Central America. And while they've been going to Los Angeles and New Orleans to some extent, the biggest community is here in New York City, probably about 200,000 in the Bronx, and another maybe 100,000 total, which are largely Queens. The English-speaking Garifuna are the ones from Belize. The Spanish-speaking, and they're more in Queens. The Spanish-speaking Garifuna are the ones here. So the Garifuna showed up in this study. And what's interesting about them is that every variant that Sri and Mariko found is novel. We've never found this. And I love this because it means that here in the Bronx, we're able to do two things. We're able to help populations that are previously understudied, and there are patients, so we can do something about it. Top of the list here, MYBPC3 causes hypertrophic cardiomyopathy. 0.0245 is the allele frequency. We have two alleles, that's 2.5%. 5% of Garifuna in the Bronx are walking around with this variant, which in a dominant form, just need one copy to be damaged, causes hypertrophic cardiomyopathy. And the penetrance, talking to Adea Weissler-Sneer, is maybe about 2/3, although that depends on people having access to care and actually getting tested. So it may be higher. Now, think of what I've just done. I'm an Irish white guy. I've just stigmatized Ashkenazi Jews, Puerto Ricans, Garifuna, by talking about the rare diseases they have because of what I've avoided calling mutations, the pathogenic variants. So one of the questions that we have, and it's in keeping with this issue of how do we deliver care sensitively to these populations, what do we do about this? So this is Sri working with two of our Garifuna community leaders at a Bronx Community Health Network event. They're with some white guy. And she's been working through Paulette Spencer and the Bronx Community Health Network to identify and work with leaders. And Tamara Watnick does this structured interview process where you get community leaders to talk what are called RAID circles, and then the interactions with the medical professionals, the scientists, whatever. It's very structured and respectful. This is where we're going with our discussion with how to impart this information to the Garifuna community perspective. And the first step was the publication, the paper that's in press. We gave the paper to each of the community leaders that were being implicated as having increased risks of rare diseases. We said, what do you think? I represent your community in a respectful way. Do you want us to take it out? We had Roma in there, and that's another very vulnerable community. So what ended up happening was everybody said, keep us in. They had some edits, and by and large, they were positive, saying, thank you for representing our community this way, and thank you for talking to us. So this can be done. So what I like the idea is that now what's rare globally is common in the Bronx. If we have 200,000 Garifuna, 5% are carrying this particular missense variant. That's 10,000 individuals in the Bronx have this very big condition. If you've got 60% penetrance, That's 6,000 of our patients who could be presenting with hypertrophic cardiomyopathy. And the nice thing about it is there's a value in early detection, right? Because you can get in there, and I'm not going to speak for you, but it looks as if there are therapies that can help to at least ameliorate the course of their condition. And you certainly don't want them turning up at a very late stage, even if your specialty is cardiac. So Adea has been very enthusiastic about the idea that she would help to understand this condition. And there's a research opportunity. She could probably get NIH funding if the NIH still exists to study the natural course, the penetrance, the attitudes of the community, and so on. And from my point of view, I love the idea that it takes advanced diagnostics to care to therapeutics that closes them. So I want to use my last slide here to ask you to consider how the New York Center for Rare Diseases can help you. If you are interested in just getting to learn more about medical genomics, if you want to build a project, if you want to have some sort of grant application, which you were hoping that you could put in, but now if you can add a little bit of stardust from the New York Center for Rare Diseases, better. You know, whatever it takes these days to get money, we will do. Okay, if you have undiagnosable patients, we'll have the ability to look at 1,200 over the next 3 years. If you want to do a genomically driven clinical trial, which is fundamentally what we're going to be doing with the NYPPC3 community here, we can do that as well. And we're already beginning to do that. I think it's very punk rock to have a rainbow these days, so it's in there. It's meant to be a tent, but
That's a symbol of nature today.
there you go. It's nothing subversive whatsoever. So right now we have a few projects which are bringing in funding and stimulating research. David Loeb is getting like a 7-figure support from the Hope and Heroes Research Initiative to study germline cancer variants in pediatric patients across the tri-state area. Myself and Joe are working on this aortopathy project, as I was mentioning. Cancer Center, we're talking about developing a profile of germline cancer risks in the Bronx and cardiogenetics, Adea's work. So this is, I would like to know if you have any ideas, approach me. And with that, I want to thank the staff that we are being able to bring in for the New York Center for Rare Diseases. Nathan is a variant interpretation scientist. Aditya is doing all the bioinformatics pipelines. Anna is a wonderful genetic counselor. Hansel is our lab technician. Carmen keeps everything going by being our project manager. And our leadership, Xiao Pang, who's one of our genetics colleagues, who's a specialist in genetically driven disorders of blood immunity, is leading our advanced diagnostics program. Melissa Watson, is head of the pediatric genetics division, is in charge of the care component. Sarah Rupp-Haft, education, control, quality operations. Now, final request, please help Shri and Tamara by taking this QR code. It's a 1-minute survey, and if they can show that they've got a few people to respond to it, it helps them with their funding, it helps them with their ongoing research. And I finished on time, which is great. Thank you all very much.
Done.
I do thank you for what I think is really fascinating part of this, the weaving of the socio-political with the genetic. I think that really helped us get a better perspective on what this weird creature called a human being is and the natural history we lived through. How do you explain, because you use the term and you can't talk about it without doing so, how do you put in context variable penetrance and late penetrance of the kind of information you're getting?
Variable penetrance versus late penetrance.
Yeah, or why not? These are things from birth. Why are they not, there on day one?
Yeah,
John, just repeat the question succinctly for the Zoom audience, because they can hear us.
Yeah, thanks for the guidance. So the question said is basically having a variant in a gene that's damaging does not guarantee that you're going to get the disease. That's the issue of penetrance. But if you have the variant in the gene from birth, why do you not have the cardiomyopathy? Why do you not have the aortic aneurysm from birth? And obviously, well, not obviously, but I think speculatively, I think some things just take time. If you have a weakness in your aortic root, and you have a bicuspid aortic valve, you've got a little bit of weakness, compared with somebody with normal aortic valve. It looks as if the turbulence over time is what causes the dilation to occur. We know that, and it has to be said that some of these variants in some of these genes can cause problems from childhood. We have 14-year-olds who are getting aortic root replacements because they are huge aorta for their age. So it's in part the severity of the damage. I think there's a time component with a lot of diseases that just has to do with wear and tear. It's very difficult to measure biologically. Or are you just stretching your
No,
arm? Sorry.
Thank you so much. I was just curious, and maybe it's too soon with what you guys have learned about the Garifuna population, but how are you then, I think, giving that information back to the communities, and what is the community outreach and then hopefully screening? What does that look like?
So to repeat the question, we have made this observation in the Garifuna community. How do we get the information back to the community? What does screening? So we're very careful. We are working with Tamara Gutnick, who has these so-called rage circles. That's her way of having structured conversations that really, it's not somebody basically being didactic and saying this is everything that's happening with you. You get people talking about heart disease in general, about medicine in general, about risks. And then you start digitating the information about cardiomyopathy, about NYBC3, about genetic issues. And it's very much community-led, community-driven, really great model. So that's the first step. And the outcome of it has to be we're in close collaboration. We jointly figure out how that message needs to go out. And we don't know what that's going to look like yet, because we're still in that process, but they're starting to record videos in English and in Spanish that will have this content that they'll be able to put out. The issue of how to do the genetic testing, it's one nucleotide change. Whether it's a G to an A or whatever it might be, but it's one nucleotide change. And it would make a lot of sense in a world with no friction to these kinds of issues. If we could develop the test locally here in our institution, because we're just in, but that's a 6-month process, and it's quite personal intensive because we have to get it qualified by New York State. So if we had an extra technician and pathology had a bit of bandwidth, we could do that. Right now, it looks as if we probably would need to do the cheapest possible panel testing. But that may be okay, because 2% of the Greally also have a tight mutation that causes dilated cardiomyopathy. And there's going to be a few people who are homozygous variants, where they have 2 NYBPC3 variants. There are going to be some people who are going to have an NYBPC3 and a TTM variant. So, do they balance out, or it's a hard scrutiny? So there's also discovery to be done once we start doing this screening, but we're not there yet. Okay, 1, 2, 3.
Well, this was an amazing lecture and very inspiring. Amanda Crone, I kind of worked within the hypertrophic cardiomyopathy program with Adalia. We kind of co-direct the program. So, I really love the idea of the bidirectional kind of feeding into research, but also into kind of clinical care. So I'm interested to kind of hear your ideas, how we can, definitely we're going to participate and try to see how we can really, provide patients and information to get new knowledge, but how can we get all those patients being tested, how we can feed them into the program so we can see them clinically and take care of them. Now that we have a program which is well established, we have options to kind of treat them with the calcium myosin inhibitor, surgery, and so on.
Once we start identifying a few people from the community and we ask about relatives who want to be tested, that's a very focused way of providing care. So a cascade testing approach. I think that for many specialties, we're going to have to start taking the testing and pushing it upstream to non-genetic specialties, as opposed to now where it bottlenecks through us in my genetics clinic. So how we deliver that is going to be a challenge, because to do a proper counseling or consent for genetic testing it's a few minutes, and you have to be comfortable. So whether we have printed or electronic materials that would help non-specialists to do this kind of consenting, I think that may be one or two.
Thank you. It was great. So I'm particularly interested about the genotype phenotype correlation, and I'm very passionate, especially about cardiac MRI. And sometimes we try to we think that we know the gene or the mutation just or looking at the phenotype, very specific phenotype, and then we encounter a PIU variant of unknown significant. How is your approach to that? You still pursue cascade screening? You want to co-segregate with the most striking phenotype in the family? You still go on with testing when you have these variants?
So if I to repeat back your question, you're interested in cardiac MRI, which I think is gorgeous. And you're asking in particular how to deal with variants.
Yeah. How you approach that? Yeah.
Ignore them. Pretend they didn't exist. 95% of them get reclassified as benign. We have no legal protection if we do something medical action with this information.
I think the question is, if you see the phenotype
Yeah.
and you test.
Yeah.
And
Phenotype.
you have the variant of uncertainty.
Ignore it.
What now?
Ignore if you ever see a variant of uncertain significance, just walk away. Do not touch it. Leave it for us. But don't you do anything regarding patient care with variant of uncertainty.
No genome-wide testing.
Well, you can talk to us. But if you've done a panel and you've only got a variant of uncertain significance, it's a good panel that's probably comprehensive. Genome-wide may or may not be of any use to you. But it could be somebody we could look at in a research. But it's variant of uncertain significance.
So you don't pursue that variant in the family to try to establish if it's a new, variant or.
That's a research project.
Yeah,
That's right.
you cannot do that.
And you don't do it, I might. Leave it to me. I can do that.
How can we send it to you?
Uh,
So what will be the process? Sorry that I'm kind of thinking of but how can that patient send it to you? What will be the practical process to do that?
Contact me through Epic or email.
Okay.
We can have a chat. It happens all the time. I get pediatric endocrinology rundowns, and patients start to flow on. Last one.
Okay, so it's kind of a question, something of a statement, but it has to do with how genetic data and Epic and Montefiore all interact
Yes.
and how it basically doesn't interact. So it's kind of like the reverse of what Aldo was saying. So I do a lot of TTR and I've done, genotype something like, a couple hundred families, but also the rest of the group is doing a lot of other genetic testing, oftentimes through commercial agencies. And we get that data and we basically it's on a PDF and it's somewhere in Epic, but not searchable, not findable. Is that normal, or is that, are there alternate models or improved models in what we're doing right now?
So the question for those still on Zoom is, how do we use genomic information more effectively within Epic when it's currently coming back PDFs that are searchable? That is unfortunately normal. There are new Epic. If Montefiore and the testing lab are both using Epic Aura, which is order results everywhere, but it's a way that you can actually have the results come back natively into the results section as opposed to the media tab, and that would make it search. We're not quite there yet because of the overload that Montefiore IT has in terms of building to spread, but we will be getting there over the next years. And then the other thing that I'm doing to make it searchable is I type in the thing into my notes, it's this particular variant which helps searchable. The other thing I was going to say, Epic has a genomics module. We're never going to use it because what it requires is that we take the patient's DNA sequence variants, put it into Epic so that Epic has access to it. Epic, as we know, sells de-identified information to insurance providers. I think with the demise of 23andMe last week and the idea that you don't have control over your data. Montefiore, I think it's fair to say we'll never have a situation where we'll expose our patient genomic information to Epic or anybody else in return for what is a pretty mediocre tool. I think I better let you go, guys. Thank you very much for your attention.
Thank you. Great. Good.
Good evening.
Welcome.
So how was it? Came in bus?
Yeah.
No, that's not. The subway's better. Where are you taking now?
Yeah, but.
Because you're in the business. Okay. Yeah, well, where is in the Bronx area? Where is located? In which area? It's Bronx. Okay. We'll take a look and see what. Here's Daniela and Alicia.
Oh, hi. Finally, we meet. Hi, I'm Annalisa. Check out the service.
Yes.
Yeah.
I was there and Daniel came to the center.
Oh, that's happening. I'm not going to the beginning.
So it's gonna continue to happen. Let's get going and everything. Let's, you guys gonna be in today?
I know.
For you, you
So
just,
I think
but I think on balance, like.
most important thing here that you make sure that you get everything you need. You can focus on what you need. Access, dispatch and everything, like, you know, and get access to everything. That's gonna take like, you know, a week or so. So, but, you know, let's get going. Let's, we can go off and we do like.
Very good, very good to try. I think it should be, right? Try to, does that make sense? But she's gonna put this on her story. Yes, I don't know how. Got it? That's
Yeah, we should be able to. And then I'm actually working on finding these people.
Hey, what's up? How you doing?
Bueno, ya veo, pues todo bajo control, ya sabemos que, ya yo decía, bueno, que quién está detrás de esto y ya entiendo que todo. The way should be. How are you?
So I think as a.
No, pero ellos para hacer que
How are you?
sacan esos o sea, pero vos no sabes, pero ellos tienen acceso. Pueden saber y decir.
Okay, great. Thank you.
Thank you.
Truly, this was awesome. So I think there's a lot of opportunities for us to collaborate. We work with Raya, just with the program here in the global medical knowledge. She does way more than just endotropic, but yeah. But I think there's a lot of things that we can do from a research perspective, but also from a clinical perspective. How we can fit those programs. Get from our clinics to you and from whatever data we can get, if it's safe to do so, and so feed the clinics to see these patients clinically and go back.
But I think from no one like so yeah, I guess I really see on.
Absolutely. For the hypertrophic, which is very common, I think, there's only like, difficult center, but I mean, none of them has like a very robust program to start. I think we can win there, but
We're actually launching it now, this month, like, our like live program just to really identify people, AI. Using the ECG and it might bring some. So he's gonna get a lot of people. I mean, some of them are gonna be like actually not and everything, but we're gonna be seeing an increased amount of people that we can be detected. This is just a pilot, so we're hoping to get into not only cardiology, but also into kind of the primary care clinics and everything, and trying to kind of really create this kind of use this technology to identify them and then like here and then of course getting testing.
That's wonderful. That's very wonderful. Yeah, access to care that's better. Genetic studies.
But anyhow, we'll be in contact and then see how we can, collaborate and help each other.
That'd be good.
Thank you.
Great. Nice to meet you.
I'm Jessica here. So,
just take order.
Ahmed is coming, he's joining us, in imaging program, is one of the.
What's your name?
Ahmed.
Oh, nice to meet you.
Yeah, so he's kind of working on some research, he's a research fellow for a year. He's similar to kind of what Danielle and then Amanda are going.
Welcome. I'm Dr. Garcia's assistant. Cardiology. So if you need anything, let me
And
know.
I think they're probably underlying,
Okay.
it's just purely,
Start today.
So and how can we?
I don't know why we get this. How should I pronounce, I mean, I always screw your name to the pronunciation. What's the best way to say it? Ahmed or Amen?
Exactly. Structure. Yeah, I know what.
That's okay.
I think that when you have an outlier.
Okay.
Especially. It's strange. Those are. Questions to you. The lab. We all know there's some both like. Those are ones that. No issues. So I'd like to have some example because there's people at this end are bad. That's really. Well, in your clinical judgment, I.
Yeah.
Why, so, as physicians, we know our gut.
I don't think it's gonna.
Those situations, they are very case. Those are what guideline. You know, conflict, like a. That's. How often? Why don't you try this? Reality is getting a sense of what would be appropriate.
But on a bigger scale, let's say 100%. Oh, yeah. That, say from genetic work, that.
You don't mind that word?
No, okay.
That becomes true.
Thank you.
Because then you're talking about. Mode of nourishment, you know. That's it. Problem with that is starts.
Yes, we do. Yes.
I go to check. So you just here, it's just change. Go through the template.
Yeah.
Anyway. Also, we revise the template that we did.
So
Based
you just
on
don't.
but now we need to like, sign on the way. People have more options and it's not always the same way. So you can say, hey, you can play around.
You have a patient. Great.
Templates, how you contact them.
We'll figure out how
You probably do
to.
it. Yes, those companies. Yeah, people launch. Question.
Yeah.
Renee, you're gonna take some breakfast with you?
Yes.
I had breakfast, thank you
Oh,
very
you
much.
did?
Yeah, give it to some hungry residents.
Okay.
I'm sure there are a few somewhere.
Our case. Management or something.
Thank you anyway.
Oh, you're welcome. It's nice meeting you.
Likewise.
Thank
you again.
I got different chat, what different chapters should be the point person. And he's happy to do it. So the way we should do this is whatever way. That we can make, not about like, what study show, I think we can discuss that, but I think we have. Definitely have something that, I think there's some of the science here.
Okay, take care.
Hey. If you're interested, we were gonna, the fellows are gonna meet the developer room now. Oh. If you wanna talk to them a little bit. Sure. But either, it's up to you too, you know.
Okay.
Yes.